Inside every single one of us right now, there's a microscopic battle going on. It's this high-stakes drama that's been playing out for literally hundreds of millions of years. Today, we're going to go deep, deep into our own DNA to uncover the incredible story of an ancient guardian, a single molecule that's working every single second to keep us alive. Let's dive in. Think about this. Every moment of your life, your body is walking this incredibly fine line. You get a paper cut. You need to plug that leak fast, right? Or you risk bleeding out. But on the other hand, if that blood clot, gets too big or forms where it's not supposed to, well, that can be just as deadly. We're talking strokes, heart attacks. So how in the world does your body manage this constant life or death balancing act? Well, the scientific name for stopping a bleed is hemostasis. It's this super complex cascade of proteins and cells that all rush to the scene of an injury to build a clot. You can think of it as your body's own emergency response team, and they are brilliant at sealing a breach. And that brings us to the million-dollar question. What tells that emergency response team, okay guys, job done, stand down? What puts the brakes on this whole clotting process so it doesn't just spiral out of control and clog up your entire system? All right, it's time to meet our hero. This is Antithrombin III, or we'll just call it ATIII for short. It's a special kind of protein, and its one absolutely critical job is to be the brake pedal for blood clotting. It's the guardian that's constantly patrolling your bloodstream, making sure clots only form when and where they're actually needed. So how does this microscopic guardian actually get the job done? Well, its method is incredibly clever, and it all comes down to its very specific shape and a little bit of molecular trickery. Okay, now this might look a little intense, but don't worry about all the crazy labels. What you're seeing here is a 3D model of the ATIII protein. Now, the most important part, the key to absolutely everything, is that flexible loop you see kind of sticking out. Just think of it as the bait in a trap. Scientists have this really cool name for it, the suicide substrate mechanism, and it works just like a molecular mousetrap. So first, AT3 dangles that flexible loop out there as bait. It's a perfect mimic of what a clotting enzyme wants to grab onto. So the enzyme takes the bait, latches on, and then, bam, AT3 snaps shut. It completely changes its shape, and in doing so, it permanently traps and deactivates that enzyme. It basically sacrifices itself to take one bad guy off the board. It's a one-and-done mission. So we've got this incredibly elegant little machine. But honestly, the most stunning part of this whole story isn't just how it works. It's for how long it has worked in exactly the same way. We're about to go way, way back in time. That's right, 450 million years. It's a timescale that is just mind-boggling. I mean, to put that in perspective, this exact protein was patrolling the blood of our ancient fish-like ancestors long before the dinosaurs even showed up. It's amazing. When scientists compared the AT3 protein from humans to chickens, turtles, even fish, they found it was all remarkably similar. The key parts, the bait, the trap, the whole structure, it's all there. When you see that level of stability over such a gigantic timescale, it tells you one thing. This design is pretty much perfect. Evolution got it right the first time and has basically had a do not touch sign on it ever since. And the stability, it goes even deeper. It's not just the protein itself, it's where the gene that builds it lives on our chromosomes. This diagram here shows a concept called synteny. Just think of it like a street map of our DNA. The AT3 gene has the same neighbor genes right next to it in us as it does in chickens. It's like the entire genetic neighborhood has been perfectly preserved for hundreds of millions of years. It's wild. But like any good detective story, this investigation into deep time uncovered a really fascinating puzzle. While the protein was super stable, the gene that codes for it had this strange difference. A piece of DNA that was there in some animals but just gone in others. Okay, so take a look here. We're looking at the actual blueprint for the AT3 gene. The black boxes are the important parts that code for the protein, and the lines are just filler DNA. Now look closely. The zebrafish gene on the bottom has an extra piece of DNA right in the middle. It's called an intron that the human gene on top just doesn't have. And this was the central mystery the researchers had to solve. Which one was the original design? Did the fish lineage somehow gain a new piece of DNA along the way? Or did the lineage that led to us and all other four-limbed animals actually lose it? So to solve this, scientists built an evolutionary family tree. Now, the key thing to see here is that they found that all the closest relatives to this gene family, they all have that extra piece of DNA. And that strongly suggests their common ancestor had it too. So if the ancestor had it and fish still have it, the conclusion was crystal clear. It wasn't a gain, it was a loss. Sometime way back, after the ancestors of land animals split off from the ancestors of fish, our lineage precisely deleted that one specific bit of DNA from the gene. And it's been gone ever since. Just a tiny little edit in our genetic code passed down for hundreds of millions of years. Okay, so we've gone from the molecular level all the way to deep, deep evolutionary time. But now let's bring this story all the way back to right here, right now, because this ancient, super stable gene has a really surprising twist when we look at modern humans. I mean, for a gene that's been so carefully protected for 450 million years, you would expect it to be pretty much identical in every single person, right? Well, it's not. Not even close. In fact, it's a hotbed of genetic variation. Get this. When researchers looked at the DNA from just over 1,000 people, they found an incredible 1,997 different genetic variants in this one gene alone. It just goes to show you that while the overall design is ancient, the little details are constantly changing. And these variations come in a few different flavors. As you can see, the vast majority are just tiny single-letter changes in the DNA code. We call them SNPs. But you also see small deletions, insertions, and other kinds of changes. Now, most of these are totally harmless. But because ATII's job is so critical, some of them can cause serious, serious problems. And this is why all this research is so important for medicine today. By mapping out these variations, scientists can start to predict which ones might spell trouble. So a change like this A414 one, it's located right in that critical bait loop we talked about. That's predicted to be damaging. It could totally break the molecular mousetrap, crippling the body's ability to put the brakes on clotting, and that could lead to really dangerous diseases. And that really is the ultimate takeaway. And the researchers said it best themselves. This journey into our deep evolutionary past, it's not just some academic trivia. Understanding the history of this gene, figuring out which parts have been protected for millions of years, gives us a roadmap to understanding how it can fail in people today. It just leaves you with this final fascinating thought, doesn't it? Antithrombin III is just one gene out of more than 20,000. Its story connects us to a 450 million-year-old ancestor and has a direct impact on our health. So you have to wonder, how many other ancient guardians, forgotten evolutionary events, and medical mysteries are still written in our code, just waiting for us to learn how to read them?